Hi everyone, today I'm going to introduce you to nuclear magnetic resonance spectroscopy. So NMR spectroscopy is a powerful method that scientists use to solve chemical structures, study large biological molecules such as proteins, and to figure out what kinds of molecules are present in an unknown solution. Okay, so today we will show you one of the applications of NMR, which is to analyze complex mixtures. So on this bench, we have prepared several everyday items which we will analyze. So we have divided them into three groups. In the first group, we have two kinds of energy drinks with and without sugar. We have coffee and we have green tea. In the second group, we have milk, cream, and soya milk. And over here, we have two different kinds of olive oil. So let's get started. Okay, so for sampling preparation, what I will do is to transfer each liquid into a tube. And I will put this tube into the spectrometer. So the NMR spectrometer is made up of a very strong magnet, which at its center is about 30,000 times the magnetic field of the Earth. Inside the magnet, there is a probe right here, which we have taken out here. So this probe contains special electronic circuits and radio frequency coils. So the tube is placed and surrounded by the coil, which will pick up radio wave signals from the sample. So the detected signals will be transferred back to the computer and analyzed. So let's get started.
Hello everyone. So the type of experiment we will talk to you today is what we refer to as a 1D proton NMR. What it actually is a simple experiment that looks at all hydrogen in your molecule. To give you an example, if we look at the caffeine, we have three methyl groups and a single hydrogen on the side. And when we look at the spectrum, it will only show us these four groups, nothing else will show up. And we will use that to analyze our mixtures. So, I will actually start with something a little bit more interesting. Let's talk about sugars. Over here, you can see glucose and sucrose, two very popular molecules to add to beverages. And they have similar structure, in fact, glucose. But what's important is that we, by looking at their spectra, this is glucose, this is sucrose, we can identify characteristic peaks. For example, these peaks, and maybe some of these could be very characteristic peaks which may be easy to identify. And in fact, if we look at our complex mixture, so two reference spectra top, this is Red Bull, And this is Red Bull without sugar. And you can clearly see that there's a lot going on, but the characteristic peaks are very easy to spot, right? And you can use these peaks to accurately quantify how much of each sugar you have in the solution. Interestingly, is also to look at the Red Bull with and without sugar, where sugar is a very dominant component, but also, for example, you can clearly see citric acid, and you could easily use these peaks to quantify it in both of the solutions. Next, I would like to talk about caffeine. You can see the methyl groups, and you can see them 1, 2, 3, as well as this hydrogen over here is this hydrogen over here. And intensity in this particular case correlates to the number of hydrogens in each functional group. And again, so if we look for that particular fingerprint, we can identify caffeine in complex mixture. So here we have caffeine, green tea, coffee, and Red Bull. I use the one without the sugar. So if you look at these 4 peaks, you can clearly see them in tea and the coffee, as well as this one over here. And you can use them to quantify caffeine. But if you look at the spectrum of the Red Bull, you can clearly see that the caffeine, despite being a very large dose, aka 3 cups worth in every can of Red Bull, it is not a dominant component. So I have to look very carefully to see that these 3 peaks and this peak correspond to caffeine in the Red Bull. But it's not the dominant species. Using same approach, we can look at another interesting molecule, lactose. Lactose intolerance. Everyone heard of it. So if we look at this molecule, again, we don't have to worry about everything. We can identify just a couple of characteristic peaks, and we can look for them in the complex mixture. So if we look at the overlay of now milk, cream, and a very popular substitute, soy milk, we can see here are your fats in the milk and the cream, and we can clearly identify characteristic peaks of lactose. And we can see that even in cream, there's a lot of lactose. But soy milk, it is very clear that those peaks are not present. As much you can see there's something here, right? You need to have the entire fingerprint always present. And you can clearly see that it is not there. And you can take even one step further. In this particular case, we're going to look at olive oil. These 2 are different brands of olive oil from 2 different locations. And the primary component of what makes an olive oil is actually the same between 2 brands. But if we look very carefully at all the small components, just like in examples of tea and coffee, there's a lot of little peaks, and those are the components that will change your flavor, and they are different between the 2 brands. And that's the power of NMR.